Hi, so you want to look at the histology of the cerebrum, the most complicated thing in the human body. It's actually not that bad under the microscope. We looked at the spinal cord last week. Similar principles. We're going to look for grey matter and white matter and see how they're different. I've got a couple of very old slides to look at. We'll look for neurons, and then we'll look at the supporting cells. There aren't very many. We're going to look for oligodendrocytes, astrocytes, microglia. There aren't that many. And again, it's going to be a slow visual look, see what we see, work our way around. You'll see it's fine. Hopefully this won't be too long. I showed you some. I just haven't got very many slides of the brain. I've got this very, very old collection that I talked about last week. So that's what we're going to use. Check this out. Can you see the sulci and the gyri in there? But also you can see this is a slide from 1937, stained with hematoxylin and eosin, which just means it looks a bit pinky and purpley. But look at the shapes there. So remember, the Brian. The reason we've got this this folded surface is because the grey matter is on the outside. The grey matter is where we find the neurons, where we find the connections. In the human brain, we may have 70 billion or 100 billion neurons, depending upon where you read, and trillions of connections. That's what gives us our complexity. That's where your anatomical knowledge is being stored. So the more surface area, the more grey matter, the more neurons, the more connections, the more complexity. So when we look under the slide, we're going to see the sulci and the gyri, the sulcus being the bit that goes in and the gyrus being the bit that sticks out. Now, I don't know anything about these slides. All I know is what's written on them. Cerebrum, HEB, in 1937. So I don't know what animal this is from. Okay, so our first job then is going to be to compare grey matter and white matter, isn't it? Our first job is going to be to switch everything on. Bum, bum, bum. All right, what we got? So I wanted to show you the whole layout on the slide because I'm not going to be able to fit it all in under the microscope. What can we see? Well, okay, so look, that's a sulcus, isn't it? Am I in focus for you guys? Like I say, I'm not sure my focal plane is quite right on these low powers. But that's a sulcus, that's a bit going in. So this is a gyrus, a bit sticking out. It's got a bit of a fold to it there. That one's a little bit torn. But these sections look exactly as they did when they were sectioned. You see these tears. Brain is very, very soft, so it's quite difficult to section. And that's one of the things about brain, is that in here, oh, that's a better bit, in here, there isn't any connective tissue supporting it. We're not finding collagen holding the brain together. That's why it's so soft, and it needs special support structures to help float it and hold it in place. Okay, how about that? That's, can you see that's a gyrus. That's a sticky outy bit. And there's the sulcus in between. So this is the surface of the brain. What do we see at the surface of the brain? Well, close to the surface, it's not looking supercellular. As we go a little bit further away, we see some very large cells, and we're seeing lots of blood vessels, capillaries and what have you in there. And then, so if that's the surface, as we go away from the surface, I have to go up a little bit, it changes and we see that layout, which may or may not be familiar to you. That may or may not look like the peripheral nerves. So what we've got is, towards the edge, we've got the grey matter. We're looking at neuron cell bodies in there and support cells. And then as we go away and we go to the middle bit, you can see how that looks different to that. That is the grey matter, and that then is the white matter. So in the white matter, we've now got axons running away from those cell bodies or maybe running to those cell bodies. But anyway, we've got axons running through the white matter, and of course the white matter is white because the axons are surrounded by myelin made by the oligodendrocytes. So that's why we're seeing these, I don't know if you can make it out, but they've got kind of got these streaky, these, it's going from kind of left to right, isn't it? We've got axons running in that direction and then meeting other axons running in other directions. Now we've got a big mass of white matter here. I've got another slide later which is going to show us the white matter a bit better. But we should start at the edge, I should think. So let's go in a little bit closer and see what we see. Now, if you read about the histology of the neocortex, of the grey matter up here, you might read that there are 6 layers. I think we can see a layer here closest to the surface. This is the molecular layer, where there aren't very many cells. There are lots of dendrites and axons here. And then as we go in a little bit deeper, we get to the next layer, which is, what's that, the granular layer. And then if we go in a bit deeper, well, that's the external granular layer, and then the cells become more triangular shaped or pyramidal, so it's the external pyramidal layer. And then as you go deeper, you've got another 3 layers as you get towards the white matter. I am not confident, let me go to a lower power to encompass a bit more of that. I am not confident at pointing at those layers. The molecular layer and the external, so the molecular layer, where there aren't very many cells, and then the next bit, where there are lots of cells, I can point at. But I am not very confident at pointing at those 6 layers, so I'm not going to. And, you know, most of the time we don't worry about it too much. But what about the cell types that we can see? What's really important in here? Well, hopefully you can already see that you've got some large cells with well-staining cell bodies. You can maybe see a nucleus and a nucleus. These are the neurons, and we're quite used to looking at neurons because we've looked at them before in ganglia and in the spinal cord. Bom bom bom. Is that better or worse? Yes, right. So we got a high density of neuron cell bodies here. Not too surprisingly for the grey matter, we've got a lot of neuron cell bodies, and that's these kind of like these big fluffy cells. And as we go deeper, sure, they do look a bit triangular. They look quite nice. So neurons are quite big, very active cells, and so around here they're going to have lots of little dendrites sticking out from the cell body, and it's the dendrites that are going to have synapses with other neurons, and that's the connections, but we can't really see those little dendrites individually here. What we're looking at, kind of the background, the stuff between the cells is the neuropil, and the neuropil is all those dendrites and synapses and connections. We would need a special stain. I have not found a section that's got that special stain that stains the neurons individually, and that's a beautiful stain, but none of the hundreds of slides that I've got have got it. So that's what the pink background is, the dendrites and what have you. And then you've got the neuron cell body, and then it's going to send its axon off, and when it gets into the white matter, it will become myelinated. And it's the oligodendrocytes that are going to myelinate these axons. Now, the oligodendrocytes are identifiable because they have lovely, they look like a fried egg. They've got a lovely round nucleus, and then they have usually pretty round cytoplasm, which appears clear to us. So we can see lots of oligodendrocytes in here, very, very round nuclei, very dark staining nuclei, and clear cytoplasm. And we can see them next to neuron cell bodies here, and those neuron cell bodies are quite different. We can often see a nucleus with a darker nucleolus inside us, and we can see that the cytoplasm of the nucleus is quite dark staining. They're quite granular because they're quite active cells, and they've got those kind of like triangular or pyramidal appearances. So oligodendrocytes and neurons, we're halfway there in our identifying the cells of the cerebrum. That's pretty good going, right? So if we roam around, if we roam around, we've got some shrinkage in places here because of the when you make a slide, you generate artifacts by dehydrating the tissues and what have you, and then sectioning, applying mechanical forces to it. But we see lots of capillaries in the grey matter. The brain is very, very well perfused by capillaries. Of course, you've got that blood-brain barrier separating the blood from the brain, but there are capillary beds throughout the grey matter. There are capillary beds throughout the brain. That's how the brain is getting all that glucose that it uses. It's very, very well vascularized. That's what we're seeing with a lot of these little wiggly shapes in here. Okay, the other cell type in here we want to try to identify are the astrocytes. Now the astrocytes, astro, star, they look like stars, except they don't look like stars to us when you just stain with hematoxylin and eosine, because everything's just a bit pink. But they got lots of processes that stick out like stars. So if we said that the oligodendrocytes look like fried eggs, they've got that dark staining, very round nucleus, and we said that the neurons are kind of triangular shaped or teardrops shaped, the astrocytes have got a paler staining nucleus and a pale staining cytoplasm, and they're a bit smaller than the neuron cell bodies. So they don't really stand out as much as either of the other two cells, which is probably why I went for it as my third choice, I guess. But I think we can see there's a candidate there in the middle. So if you use a silver stain, you can make the neurons stand out really nicely. If you use lots of different stains, you can get cells to stand out really, really nicely and show you different things. Hematoxylin and eosine is a very general stain, which gives you that pinky general colour and then the purpley colour of the nuclei. And different cells will take up the stain in slightly different ways, which gives us slightly different looks. So let's get deeper. Right, there's the white matter. So let's get in there. Oh, there's a good mix of cells. So this is very much cerebrum, very much grey matter. We can pick out some neurons. We can pick out some oligodendrocytes. I think we can pick out some astrocytes. And the other cell type is the microglia. Oh, there's one there, I think. The astrocytes, what do they do? What are they there for? Well, I said that the brain doesn't have any collagen in it, really. It doesn't really have any connective tissue into it. So one of the jobs of the astrocytes is to give mechanical support to the very, very soft brain. They're also involved in helping shuttle things between the blood and the interstitium around the neurons, the metabolites and large molecules and that sort of thing. And the astrocytes are probably involved in repair processes and lots and lots of jobs. The astrocytes seem to do lots and lots of jobs, and I expect as we continue to research this, we'll find that astrocytes do more than we thought. If you go and read about astrocytes, you'll see that they're very busy. But the microglia, we've got neurons, and then we've got glial cells. So the glial cells are the support cells. So the oligodendrocytes and the astrocytes are glial cells. It comes from the word meaning glue. Glues everything together. Pretty cool, right? The microglial cells, can you see those nuclei? There are some nuclei in there that are longer and flattened. And if we looked at the cells, we would find that they were smaller cells and they're probably a bit longer and flattened. Those are the microglial cells. And the microglial cells are like the phagocytes of the central nervous system. So if phagocytes elsewhere in the body are the big eaters, they go and munch up stuff that shouldn't be there and take it away. That's a big role for the microglial cells. They eat up things that shouldn't be here and get rid of it. And that means that they do actually change their shape when they're in different modes and doing different jobs and what have you. I can see, of course, I'm talking about these very happily in my field of view, but can you see them? There we whoop. See that flattened nucleus right in the middle there? And then I saw another one. Where was it? Where are you? Oh, there's one in the middle there. So you see these flat, skinny nuclei, they stand out. They're quite different. Yeah, and surprisingly, that's it. Is the histology of the cerebrum the simple part of the cerebrum? Oh no, well, that's the grey matter. We still got to do the white matter. But the white matter is the same stuff, it's just going a different way. But you've got your microglial cells, your astrocytes, your oligodendrocytes, and your neurons. So if we zoom out, we are seeing that mix of cells. So there's our grey matter, and as I scroll up, you can see that arrangement of grey matter shift, and that looks different now. Now we've got those streaks from right to left. So that's the white matter. All of those neurons in the grey matter, that's where their axons are running to, and their axons are now bundled together in the white matter as tracts. So their axons are myelinated by those oligodendrocytes. You might have one oligodendrocyte that it wraps around 5 or 6 axons and travels a certain way, and then you have another oligodendrocyte doing the same thing, and so on and so on. And we talked about myelin and the diameters of axons when we were talking about peripheral nerves, and the two things are really important. So you need a large diameter axon to propagate action potentials. That's the signal that runs along the axon from one place to another. That's the purpose of the neuron, right? You need, the larger the diameter, the faster the action potentials will run down the axon. Or you can surround the axon with myelin and have little gaps, the nodes of Ranvier, and the action potential will jump from node to node. So you can have a much narrower diameter axon, which means you can pack more in, which means you can get more complexity, which is good. And then you can also make your action potentials go even faster. So that's why we see the white matter. And the white matter is in the center. Like I say, the grey matter is around the outside, and that's where we have the neuron cell bodies. So then the white matter is running through the center of the brain to get down the brainstem and off to wherever it's going. So the white matter is in the middle. And we see the opposite in the spinal cord. We see the grey matter running down the middle and the white matter running around the outside. So this is all white matter. So these are axons running across the screen, and the other cells we see there are the same glial cells supporting them. Pretty cool, huh? And yeah. So we're not seeing neuronal cell bodies here, we're just seeing the cell bodies of the support cells. And it's looking very nervy across here. When we looked at the tissues, I said, there's some capillaries there. By looking at lots of tissues and taking time to look at tissues, you just get used to what tissues look like. And there is the cut edge of the section. So that's all the neurons there have been cut through. Very nice. And like I say, that section is from 1937 that was made. And yeah, gyrus, sulcus. It still works. It's probably in exactly the same condition as when it was first made, and I'll put it back into its box. But I have got another slide for you. When we were looking at the peripheral nerves, I think they were stained with osmium tetroxide, which is great for looking at lipids, so we could see the myelin in the peripheral nerve. We could see those rings, and it was very, very distinctive. Silver stain is great for staining neurons, so then you could see all those dendrites and axons and what have you. I've got some slides here. Again, I know nothing about them other than what's written on the slide, and they're big. This one says visual cortex. And look at the size of that thing. Also, look at the colour of it. I think this is probably vile stain, which again is going to stain the myelin in the central nervous system. So the black there is the white matter and the grey matter is around the outside. So I don't know if you can see, we've got some cut edges, but we've also got the hints of some gyri and sulci there. So if we look at this under the microscope, the dark staining should be the myelin, should be the white matter, so you can see some of that arrangement a bit better. But I don't think it's going to look very good at high power. I think it's probably a fairly thick section, a bit of a chunky section. What do we got? Okay. What am I doing for focus? So that is the surface of the brain. So that's the surface, that's the edge. And we can... if I go down here, so we're looking at the surface of the brain. See, there's a cut edge. So that's a square edge where the tissue was cut from the brain. Again, I've no idea what brain this is from. So that there, if that's the surface, then that is a sulcus. All right? So that's a gap between the gyri. And we're seeing some blood vessels in there. Ooh, so are the blood vessels staining black. I don't know much about vile stain, I'm just guessing that's what it is. Maybe I should have looked this up beforehand. Anywho, so maybe we're seeing lots of blood vessels in there with those streaks. Anywho, so if that is a sulcus, then this is the outer edge of a gyrus. So the grey matter is not very well stained there. It's a very light brown. I think we can see the nuclei in there. The nuclei are picking up the stain. And then as I go in, you can see those streaks running towards the centre, and then it gets very dark. So the very dark patch, that's the white matter, so myelinated axons. And that streaking pattern is beautiful. What we're seeing there is we're seeing the neuron cell bodies in the grey matter sending their axons in towards the white matter, and you can see that process of myelination. And then as those neurons, those axons all get packed together, that density of the stain gets really dark. Oh, very cool. Right, if I go to a higher power, I've got a good chance of being in focus. That's pitch black. Right. Super black. So, go to the edge. Oh, that's too bright now. Go to the edge. There's the grey matter. Not really staining. We've got some nuclei staining. Looks like we've got lots of blood vessels staining. That might give you an idea of the capillarization. As we go in towards the white matter, we can see the myelin being stained. We can see some of those streaks. We can see some blood vessels in there being highlighted now. And as we get closer to the white matter, it gets darker and darker and darker. And like I say, if I go to high power, yeah, there seems it's a bit thick, maybe looking a bit blurry. And because that stain is staining the myelin, and there's so much myelin in there, we're maybe missing the detail a bit. So there's the white matter. Slide back out towards the grey matter. We're getting too bright. And we're just looking, and we can see even the cell bodies aren't, the nuclei even aren't staining. It's interesting how the camera picks up that brightness much better than my eye does, that change in brightness. And there's that surface layer then there. So we can We can maybe see some of the texture in the surface layer of those connections. Anyway, I imagine these sections are pretty old as well, but I don't know how old they are. But I think that's it for cerebrum. Oh yeah, we've got quite a bit of thickness here. Look at that. Boom, boom. You can see a triangular cell in the middle there, and then come back out to that one. Thickness doesn't really help when you're looking at histological sections, but it does help. It is required for some stains. Boom, 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 boom. So this is probably, yes, a few cells thick. Right, I'm just rambling now, aren't I? All right, let's go back out again. Boom. So again, like much of the histology we've been looking at, and particularly I think with the nervous system, you have to imagine a lot of the microscopic and cellular stuff, and by looking at the histology, looking at the cells, looking at the organisation, hopefully that helps your imagination, or you have to imagine less, but that's the organisation of the cells in the cerebrum. Neurons, oligodendrocytes making myelin, astrocytes doing pretty much everything, microglia clearing up after everybody, lots of capillaries, and the white matter being the tracts of axons going off to somewhere else and carrying those action potentials to wherever they're needed, whereas the grey matter is where you have those connections. It would be lovely if I find a slide that shows neurons that's got a silver stain, then I'll add to this. But I think we'll lay off the nervous system for a while and we'll go and look at another tissue, which is, I am tempted because I've got a cerebellum section. We'll see what happens. Anyway, I hope that was useful and interesting. See you next week.